The overall goal of this novel separations technique is to fractionate native samples with high recovery and resolution while maintaining non-covalent macromolecular interactions such as protein complexes.
This method can help answer key questions in the study of native macromolecular complexes, such as complex composition and stoichiometry.
The main advantage of this technique is its high recovery and low sample dilution, unrivaled by any other native separations method.
Begin with assembling the casting system. The first step is to use a hot blade to cut the tip off a 20 milliliter serological pipette. Cut orthogonally 10 centimeters from the top of the dispensing tip. Then, clamp the cut pipette tip down to a support stand. Next, cut a 3 centimeter length of tubing to attach to the tip. Stretch one end of the tubing over the tip and attach a flow control valve. Connect the valve to the dispensing tip of a gradient mixer using another piece of tubing. Keep the valve open for now. Next, put the gradient mixer on a magnetic stirrer and load stir bars into the mixing chambers. Now, test the system for leaks using deionized water. Then, dry the system by blowing air through it. Cover the top of the pipette section with 2 layers of tape. Then, puncture a hole through the tape layers